medications undergo years of development and safety testing before ever being given to humans. And once they're approved to move into human research, the drugs are still being observed for how well they work and any side effects they may cause. This practice helps ensure that we're receiving the best possible product, which when taken appropriately won't cause us harm. This model has seemed to work well, but Sophie Zaaijer says that most of this research is overlooking one major variable: skin color.
We have this extreme diversity of skin tones that really differentiates human populations. I all of a sudden realized, what do we know about skin pigmentation in relationship to drug testing and to the interaction with drugs? This is where we started identifying, hey, first of all, there's very little information about this. So there's very little research started already in the 70s, 80s. But then you see over time that it's very sparsely investigated.
Zaaijer is a molecular biologist and consultant for diversity, equity, and inclusion in research and development, as well as clinical trials. After the connection between melanin and drug safety was first realized over 40 years ago, people mostly forgot about it. The understanding of how our bodies metabolize drugs was never impacted, a mistake that's putting countless lives at risk. In general, after a drug enters the bloodstream, it's usually processed by enzymes, which reduces its effectiveness. But this conversion is taken into account during the research process. However, Groen says that the amount of melanin a person has can create even more change, which is almost never considered.
Melanin can kind of act as a sponge in the skin to take away even more drug from the bloodstream. So depending on those enzymes in the liver and the melanin in the skin, together they kind of determine how much drug is becoming available to actually reach its intended target. So this is very dependent on the type of drug, but for some drugs, melanin can have a very important effect in changing the availability of the drug for reaching the target.
Groen is an assistant professor of evolutionary systems biology at the University of California at Riverside. The fact that melanin attracts drugs isn't the only issue. After they bind together, the melanin slowly releases it back into the body.
So let's say drugs are going to the skin and bind to melanin, then that diminishes the availability in the short term of that drug. But then over the longer term, if that drug is then being released by the melanin, then you might have a more prolonged action of that drug.
So they have these time course experiments where they looked in an animal model. They show that there is more attraction to these melanin-rich regions in the body, which includes the skin, the eyes, and also hair. And then they see this slower release of the compound in the bloodstream over periods of time. But I think much more research is needed to really carefully mimic and study this, how this actually works.
Researchers have been using this phenomenon to their advantage when creating eye medications, and it could make treating skin conditions more effective. But the dangerous downside to this process has been underestimated. The first concern is that at a research level, this effect could mess with the dosing studies. It could appear that the drug is inconsistent when in reality, it's a difference in how much of that drug is available for the body to use. Down the road, this can lead to doctors under or over prescribing medications to people with pigmented skin. But the second question is, if the melanin slowly releases the drug back into the body, when do you give a second dose?
So the timing of the dosing is important as well, because that's then, that also shifts over time because you get this absorption and release, slow release. So it's timing of the dosage and the dosage itself for the treatment of the intended target.
In an effort to rectify this oversight, Congress recently amended the Federal Food, Drug, and Cosmetic Act to require diversity action plans for certain clinical trials of drugs, medical devices, and biological products. This means that the research sponsors will have to set diversity goals for their participants based on sex, race, ethnicity, and age.
And so they're working on that in order to get that to basically recruit more people from African descent or Asian descent or Hispanics. But there's a huge gap but in preclinical, there's no attention for it. And that is exactly what we would like to illuminate, because if you recruit the people in clinical trials, if diverse people, they need to know the safety and efficacy data from preclinical to make an informed decision. And at the moment, that cannot be given because nobody is looking at it in preclinical.
However, there are current technologies available to make this happen. It's just a matter of the industry deciding that they want to. Zaaijer and Groen recently published an article in the journal Human Genomics that aims to light a fire under the scientific community to finally take this information seriously. They break down why we should care and offer tangible actions researchers can do right now that will make a big difference. Zaaijer says these tests can even be done in a petri dish.
And so now it becomes interesting because you can test it in the laboratory to understand the relationship between drugs and pigments, to understand how this dynamic could be interrupted or change. And you can use this to make clinical decisions, what you measure and at what frequency you measure it. So it's really informative for clinical decision making to do the initial test in a petri dish.
Another innovative option is using 3D skin models. Their development has been pioneered by the beauty industry in an effort to test products more accurately while also moving away from animal testing.
So L'Oreal was a front runner. Actually, most of the real beautiful scientific papers describing the skin and pigment in the skin are done by scientists working at L'Oreal. But through that innovation, actually now companies have been formed that actually offer these 3D skin models with and without pigments for people to purchase and actually use in their own labs. And it's really a turnkey solution. So you put your order in and you get it with all the media and everything. You can start right away, which is so important because otherwise, every pharma company, let's say, would have to develop their own skin models and have to standardize it to make it reproducible, so to create reproducible results. But because these vendors now exist, creating these 3D cell models, pharma labs can adopt them right away and actually test whether their drugs has indeed affinity for melanin and start investigating indeed what the kinetics, how it affects the kinetics of the drug in the body.
Once they know these effects, they can adjust the clinical protocols and create safer, more effective products for people with higher levels of melanin in their skin. However, Zaaijer notes that accounting for melanin is just the first step. When doing this research in human cells, she says that scientists really need to be reporting the ancestry of these samples.
There's a lot of genetic variants that contribute to whether a skin is pigmented or not. So we don't really understand how it genetically works. So therefore, it's super important to have genetic ancestry associated with the skin models used in the laboratories so we can understand how the drug kinetics is related. Eventually, maybe in a couple of years, understand how the drug kinetics is related to genetics and basically variants, and that we can now start to predict it in the future better.
Which will help streamline the research process. And while AI can be an incredibly useful tool in combing through all of this data, scientists need to first broaden the data points they're looking at.
I don't think we are giving sufficient variables or we're measuring sufficient variables for potential algorithms to extract how a drug works differently in a person of Northern European descent versus a person of African descent. And we need to work towards that, giving all the variables, genetics, how does a drug interact with melanin from skin models, the metabolites of the liver. All these variables need to be part of the equation to truly get to treat people in an individual way, so working towards precision medicine.
And as she mentioned earlier, Zaaijer believes that the action that can have the most immediate impact is fairly simple. Scientists need to start documenting the specific ancestry of the cells used in research.
If you use the cell of Northern European descent, report it. That's now not happening. If you use a cell of Northern African descent, and we want to be as specific as possible, report it. And so, because once we start reporting, we have data, we have this information we can use to start seeing patterns. We can start using that indeed in algorithms that we are using increasingly. But at the moment, in terms of diversity and acknowledging that there's different human populations, there's not enough reporting in the earlier stages and later on clinical trials either. And once we start reporting, we can start to see patterns.
While this type of detailed reporting is part of the scientific process, Zaaijer says that for some reason it's been ignored when it comes to human diversity. Both Zaaijer and Groen hope their advocacy reaches the patients and helps them realize the questions they should be asking.
For example, when they are considered for participation in a clinical trial, they could ask the doctors like, hey, before you give me this new drug that is in development, has this been tested on cell models that are representative for my ancestry? So right now, if people are of like European descent, then in most cases, the answer is yes, it's been tested on cell models from European descent. But for people originally from like sub-Saharan African descent, then it's often the case that such drugs have not yet been tested preclinically on cell models that also have origins in sub-Saharan Africa. So that's something that people should, I think, really ask their doctors, like, hey, is this experimental drug like really safe for people of my ancestry?
Groen believes that having more people ask these questions will convince the pharmaceutical companies to start considering this diversity in their preclinical research. And at the very least, it gives the patients better information to decide if they're willing to accept the potential risks of the drugs they're taking. You can find more information about all of our guests on our website, ingoodhealthpodcast.org. For more behind the scenes, follow In Good Health Pod on Facebook, Instagram, and X. Our writer-producer is Kristen Farrah. Our executive producer is Zaaijer. I'm Elizabeth Westfield.